Hello and welcome to Module 3, Setting Up of a Clinical Trial and Preparing a Clinical Trial Protocol. My name is Jannik Stemler. I am a physician scientist at University Hospital of Cologne and also affiliated with CECAD Aging Research at the Institute of Translational Research. We start off with a citation from the International Conference on Harmonization of Good Clinical Practice on the study protocol, which is a document that describes the objectives, design, methodology, statistical considerations, and the organization of a clinical trial. Usually, the protocol contains a description of the background and rationale of the trial, but these topics can also be covered in other documents. And moreover, the term protocol refers not only to the protocol itself, but also includes its amendments along the way. Since the protocol is the basis for the proper conduct of a clinical trial and a reference book for all essential questions, it is one of the most important documents of the trial, and so to say, the central element. However, specific details can also be covered by other documents, such as the laboratory manual, the monitoring manual, the safety handling plan, or the statistical analysis plan. Changes to the study protocol must be documented by the sponsor, and if necessary, approved as an amendment by the legal authorities and ethics committees. The study protocol provides the basis for quality assurance, as it includes all the details on how the trial needs to be conducted, that comparable data is produced. To whom is the study protocol important? So for all parties involved in the study conduct, such as the investigator, the study nurses, the pharmacist, and the laboratory, the study protocol is a major document they have to refer to during the trial. But also for the monitor, the data managers, the data safety, and the data monitoring committees. Furthermore, funders, such as pharmaceutical companies or public funding bodies, for example, European Union funding programs, the German Cancer Aid, but also private funding bodies, such as certain foundations, can be involved in the study protocol. And of course, it's also important to the ethics committees who review the study protocol prior to its approval, legal authorities, and inspectors and auditors by these authorities. The study protocol and its amendments are usually written in English, which is the pivotal valid language internationally applicable for clinical trials. It is also allowed to translate or adapt the study protocol to use it as a working version, but only the approved English version is valid and binding. In most cases, however, a summary of the study protocol in the national language is available in the respective countries. So we are now going through the elements of a study protocol, and basically we start easy off with the cover sheet. The study protocol consists first of a cover sheet and a synopsis, which contains specific information. We start with the OIDRA CT number or any other identification number, depending on the country-specific regulation for clinical trials, and other additional registration codes which apply to the specific trial. In addition, the sponsor code has to be written on the cover sheet, as well as the study title and a short title of the study. Also important is the indication of the version number, the number of amendments, and the date of the study protocol version. If applicable, the synopsis should also include a pocket version, which summarizes all relevant aspects of a clinical trial, and I'll go into detail on this pocket version later on. Second, the contact details should be included in the first parts of the study protocol. It should include the names and titles, addresses and telephone numbers of the sponsor or the sponsor representative, or any other persons who are authorized to sign the protocol and the protocol amendments for the sponsor. It should also include the contact details of the sponsor's medical experts for the trial and of the monitor, if other than the sponsors. but also the contact details of the clinical laboratory and other medical or technical departments or institutions involved in the trial have to be listed. In case the study is conducted as a multi-center trial, the contact information of the coordinating investigator has to be indicated. If the study is conducted as a monocentric trial, then the contact details of the investigator or the principal investigator have to be given. Usually, the study protocol contains an additional sheet with the contact details of all participating investigators and trial sites. Because the additional sheet is often a loose sheet that is not a permanent part of the protocol and changes are done more frequently, no amendment is necessary for changes in the investigator's contact information. Moreover, the study protocol comprises tables and flowcharts which illustrate the type and design of the trial, including the procedures and stages. These illustrations should always correspond exactly to the wording in the study protocol and must be understandable on their own. And they are usually the parts of the study protocol investigators most often refer to because they give an easy overview on the conduct of the trial. The objectives and the purpose should be defined and phrased clearly. So the background information, including a brief description of the medical issue and current treatment options, should be included before starting off with the details on the conduct of the trial. And a summary of the findings from non-clinical, preclinical, and other clinical studies can be addressed here. Also, a summary of the known potential risks and benefits of the investigational medicinal product can be included here, and furthermore, reference to the relevant literature and data are included. Also, a statement that the clinical trial will be conducted in compliance with the protocol, good clinical practice guidelines, and the applicable regulatory requirements should be included here at the start of the protocol. The study protocol should provide a description of the rationale, including the reason for the study and for the population to be studied, a justification for the route of administration, the dose regimen, and treatment periods. This mostly applies to later stage clinical trials, so phase 2 or phase 3 trials, where these details are already defined after preclinical and phase 1 studies. Furthermore, the motivation for the selected study design and study medication should be included here, as well as a risk-benefit assessment, which includes a detailed description of all study-related risks as far as known, so also including potential risks, as well as the expected benefits for the study population. And this, of course, should answer the question that the study is ethically justified. We continue with the inclusion and exclusion criteria, which are a central element for the conduct of the clinical trial. They must be described clearly and with little scope for interpretation. The study protocol should comprise measurement categories or diagnostics to examine these criteria. And also the in- and exclusion criteria have to be in accordance with the contraindications of the investigational medicinal product. The objectives and outcome measures, this is what we call endpoints during a trial, should be listed, including the primary and secondary endpoints. Usually, a clinical study contains one primary endpoint and several secondary endpoints, as well as exploratory endpoints if they're needed or if they're deemed justified. For each objective, which we listed before, one corresponding endpoint should be included. Then the study protocol should include timelines, such as the expected duration of subject participation and the description of the sequence and duration of all trial periods, including the follow-up. This includes specific timelines, such as when is the first patient in, when is the last patient out, and when is the end of the full trial, etc. Another important point in the study protocol is the specification of the stopping criteria or criteria for patient withdrawal. These have to be defined for the entire study as well as for individual participants. The subject withdrawal criteria define when and how a patient withdraws from the trial, and the type and timing of data to be collected for the excluded patients. For example, if a patient withdraws consent, it should be specified if data which are already available before withdrawal of consent can also be analyzed, or if this data should be deleted. It should also be included whether and how subjects are to be replaced and followed up. And other scenarios could be that the entire trial might have to be stopped due to safety reasons. The next important topic is trial medication. So the trial medication should include all trial treatments, the dosage and the dosage regimen for the investigational medicinal product, which means not only the dose itself, but also the route of administration and the number of dosages per day or per week, as well as the dosage form, so oral, IV, etc. And the packaging and labeling of the investigational medicinal product are also to be described in the study protocol. Moreover, the protocol specifies the administration of the investigational medicinal product, its storage and logistics. These details could also be included in a laboratory manual or in the investigator's brochure on the IMP. And then it also includes the documentation, accountability procedures for the IMP, including the placebo, if applicable, and the comparator, if applicable. We are now going into details on the dose of the IMP. The protocol should provide detailed justification for the dosage and the duration of the therapy, and adjustments of the dose during the trial must be explained in detail. For better understanding, the visit plan should be presented as a graphical flowchart in addition to this description in text form. And this is also a part of the study protocol, which is very central for investigators to refer to gain overview on the type of visits and the conduct of the clinical trial. Furthermore, the protocol includes a description of the treatment of subjects by listing the treatments to be administered. Those should include the product names, doses, dosing schedules, administration route, and treatment periods. To avoid the withdrawal of the subjects, any additional medications and treatments that are not part of the clinical trial should be listed here, if they are permitted before and during the trial, as well as procedures to monitor the subject compliance. And these should be indicated here at this stage of the protocol. To assess the efficacy of the treatment, the study protocol should specify efficacy parameters, methods and timing for the assessment, and recording and the analysis of those efficacy parameters. So these are very detailed informations that should be included in the study protocol and should be thought about very thoroughly before giving the protocol for approval at the healthcare authorities and ethics committees to not have to amend those efficacy parameters and the way they are assessed. A central element in the study protocol is the assessment of safety. For this purpose, safety parameters, methods, and timing for the assessment and the recording and the analysis are defined priorly. Furthermore, a procedure for eliciting reports of and for recording and reporting adverse events and intercurrent illnesses has to be established before starting the trial. And this is usually one of the most important requirements by healthcare authorities and ethics committees. Moreover, the time and duration of the follow-up of the subjects after an adverse events have to be specified here. For the documentation and follow-up of adverse events, all events and reactions have to be defined precisely, including their severity and expectedness. So adverse events have to be differentiated from adverse reactions, from serious adverse events, from serious adverse reactions, and from suspected, unexpected serious adverse reactions. Moreover, adverse events of special interest must be listed and classified using the CTCAE grading scale, which stands for Common Terminology Criteria for Adverse Events. And this applies specifically for medications which are known to have certain side effects from preclinical or phase 1 clinical trials, when certain side effects may have occurred, which are specific to the IMP. And furthermore, the time for the acquisition of those events should also be defined within the protocol. The monitoring should provide the verification of source data or should be risk adapted. In addition to the description within the study protocol, details are specified in the monitoring manual, which is a separate document and less stringent. The data monitoring committee is responsible for monitoring of the trial progress and safety data, but also monitors efficacy targets. For instance, in the context of result evaluation of interim analyses, this is something that could be applied here. And the DMC, the data monitoring committee, has the responsibility to consult the sponsor about the continuation, modification, or termination of the clinical trial regarding interim evaluations or safety analyses. A central topic of the study protocol are statistics. The protocol comprises the description of the statistical methods and the timing of any planned interim analyses, and of course also details on the final analyses. The number of subjects planned to be enrolled is specified in the protocol by defining for a multi-center clinical trial the number of enrolled subjects anticipated for each trial site and the reason for the chosen sample size and its clinical justification. Moreover, the level of significance and the criteria for the termination of the trial have to be indicated from a statistical perspective. And the procedure for reporting of any deviations from the original statistical plan and the selection of subjects, all the eligible and evaluable subjects, should be described in this section of the protocol. One important aspect is data protection. It should involve all specifications on this part of data protection regulations. Very important are explanation on pseudonymization of participants, or also anonymization if that's possible, and regulations on data transfer to other countries, to other investigators, etc. It also includes information about data management, defining which database system is used, if a paper-based or electronic case report form is used, and a description on validation, data protection, and data backup. This should be maintained in this section of the protocol. Usually this part also includes references to the certain manuals, which I have mentioned before. In another part of the protocol, the sponsor has to ensure that access to source data and documents is described. The investigator or institution will permit trial-related monitoring, audits, review by the IRB or the ethics committees, and regulatory inspections by providing access to source data and documents. Without this section, a study protocol usually cannot be approved. In addition, the study protocol comprises parts describing quality control and quality assurance, ethical consideration in relation to the clinical trial, data handling and record keeping, financing and insurance, if that applies to study participants, and this is included if this is not addressed in a separate agreement, and the publication policy if this is not addressed in a separate agreement among the investigators. Furthermore, there are supplements. Since the study protocol and the study report are closely related, further relevant information can be found in the ICH-E3 guideline structure and content of clinical study reports. In the next section, I'll show you which aspects have to be considered when preparing the study protocol. But first of all, who is responsible for the preparation of the protocol? This responsibility lies with the study leader, which can be the sponsor himself, or an institution or a person that is commissioned by the sponsor, such as a clinical research organization. And these institutions or persons are then finally responsible for the preparation of the study protocol and have to assure that the conduct of the trial is well prepared. So usually the study protocol is prepared by several parties and a well-organized coordination is required. And of course, those involved parties can be the medical study leader or the medical writing team, the biometry team, the project management, the safety management, the monitoring and the data management committee. So there are so many parties involved that an overview and a very well-organized coordination is pivotal and crucial for a well-prepared study protocol. This is what I've mentioned before. We jump to the next session. Prior to the internal finalization of the study protocol, it has to be reviewed by the investigators involved, by study assistants, pharmacists, the laboratory team, the data monitoring committee, and the sponsor. The approval by the sponsor is required in any case, and the study leader and biometrician should sign the protocol before it is submitted to the ethics committee and the legal authorities. The compliance with the protocol is required, and deviations or changes of the protocol are only allowed in agreement with the sponsor, and if required, also by the regulatory authorities after internal review board or ethics committee approval and a favorable opinion of those committees, of course. An exception is when the changes are necessary to eliminate an immediate hazard to trial subjects, or whenever they are present, or when logistical or administrative changes, such as the change of the monitor, phone numbers, and stuff like that, are very time sensitive. The investigator or person designated by him should document and explain any deviation from the approved protocol. In case of protocol changes due to the protection of trial subjects from hazards, the implemented deviation or change and the reasons for it, and if applicable, also the proposed protocol amendment should be submitted to the IRB and the ethics committee, again, for review and approval, to the sponsor for agreement if this is required, and to the regulatory authorities if this is required. So now we are going over these protocol amendments. According to the ICH-GCP guidelines, a protocol amendment is a written description of changes to or formal clarification of a protocol. Modifications of a study protocol are only allowed when a protocol amendment is submitted, as I pointed out before. And a protocol amendment is a written description of one or more changes to or formal clarification, as I pointed out before. Those amendments can be non-substantial, and that includes just minor changes in some aspects of the protocol, or substantial, which may have an impact on safety, physical or mental integrity of the subjects, the scientific value of the trial, the conduct or management of the trial, and the quality or safety of any IMP used in the trial. So those substantial amendments would include a change of the sample size, as well as change of administration route or something like that. And those amendments are really important and should be planned well, and also agreed upon by all investigators involved. Here we see an overview. You can see the different steps that have to be considered after modification of the study protocol. So after the protocol has been assessed according to national regulatory requirements, modifications of the protocol have to be in agreement with the sponsor and investigators and need to be prepared as written amendments. Usually, amendments comprise a tabular list which contains amended, deleted, or inserted passages and the respective justification. However, the form of the amendment depends on the national requirements, of course, and moreover, it has to be considered if an adaptation of the patient information or CRF is also necessary due to this protocol amendment. Only after the assessment of the amendments according to the national and regulatory requirements, the amendments are valid from this time point where the approval has taken place. And once the amendment is approved, it has to be distributed to all stakeholders and investigators. In particular, it has to be considered if already recruited patients are affected. And in this case, the informed consent must be renewed for all current trial participants and also those that already had been followed up. Since the protocol is such an important document, the theory has to be transferred to the daily routine at the trial site by training of the study staff, which will be covered in this section. So the transfer from the theory of the protocol and the sometimes strict requirements to the practical work during the conduct of a clinical trial at a site. So to ensure that the study group is well informed, preparing a pocket information can be helpful, and I mentioned this earlier. So this could be a small card or a brief overview on that summarizes all important information about the study, such as the visit schedule, the in- and exclusion criteria, the most important ones at least, the requirements for the storage and dispensing of the trial medication, as well as the most important contact details, for instance, from the monitor for the serious adverse events management, from the laboratory monitor, sponsor, etc. So this is really useful so that study nurses and investigators can have a brief look if they are talking to a eligible or potentially eligible patient on the visit schedule to inform them as well as possible. Of course, these pocket infos do not replace the informed consent form or the protocol itself. Furthermore, manuals, for example, safety manuals, are helping to implement the study protocol at the trial sites, as well as logs, such as the screening log or the inclusion log, provide assistance with the documentation and also help to maintain the overview on the conduct of the trial at a specific site, and can also help to detect gaps during screening or trial participant inclusion. Of course, this additional information must be based on and be in compliance with the study protocol. And this almost concludes my presentation. I'd just like to quickly summarize the main points and take home messages. So as you already have heard several times during this presentation, the study protocol is one of the most, or maybe the most important document during a clinical trial, because it ensures that the rights, safety, and the well-being of patients and participants are protected, and that the results of the clinical trial are valid and credible. Most important, the objectives of a clinical trial can only be achieved in compliance with good clinical practice and ethical principles. This should be the main takeaway message from this module 3. There's more information in the International Conference on Harmonization of Good Clinical Practice, Chapter 6. And this full chapter is on the clinical trial protocol and protocol amendments. But in this module, you got a brief summary by myself. Thank you very much for joining today. And for questions and remarks, please contact the given email address. Thanks for your attention.